Hi, welcome to Scanning and Grinding, where we try to answer the question what's going on inside the fossil? Because the outside isn't necessarily the most interesting part of a fossil, and you can usually only see the outside or the inside, rarely. It's rare that it would be both. You'd have to have a perfect loose fossil. So, the lowest tech and most widely applied, and you could even say maybe best way, is sectioning. So sectioning is something you would do to like a piece of meat or a little. You could section a mouse brain and then look at a little thin slice of it on there. You make thin slices, but of rock, and then you can see what's going on in the rock. So these are thin slices of something like limestone, and look at this. It's got little corals in there, oriented so we can read them. There's a little coral there. This is another little coral that's a little bit crushed and filled in. You got little bits of gastropods in there, little bits of bivalves in there, little bits of algae in there. All kinds of really neat stuff. And if you're just looking at it regularly, you would say, it's rock, it's limestone, there's a little bit of crud in it or something, but you can actually be very precise about that. And this is how they call different types of limestone by their proper names and different types of shale. It's by the average size of the grains, what's cementing it, what is being cemented, how well cemented is all the stuff, and yeah. So these names would then tell you a little bit of the geology, like the science of, well, how did this thing come to exist? So pretty useful. These are all super thin slices, and I think they're all shot with a polarized light filter, which makes these beautiful little rainbow colors appear on some of the items. Really neat. And they're showing you differences in the way crystals are aligned inside the structure of the fossil. Very useful if you're trying to dissect and figure out how did this fossil form when it was alive, because the orientation of crystal grains can tell you a lot about that. Okay, so the procedure for one procedure for reconstructing a three-dimensional specimen is serial grinding. And so the way the pros do it is they will have a little mount like this, and you can screw it together. And as you screw it, you're basically screwing in the center, and you're pushing up the little section. Now they got the section aligned, and they've probably embedded it in some kind of resin, something like that. Or in this case, it was in a nodule, and they've shaved down the nodule to just get the parts that they think are interesting on there. And you do that with rock saws. There's no special tech involved. And so then what you do is you take that thing, you flip it upside down, and you just grind it on a spinning grinder like we will use in the lab, or just with sandpaper. You just go back and forth. And because the height of this is set on the side and it's actually marked out, you can make sure that you're grinding in particular increments. You can grind, 10 microns and then stop and then take a picture of it and then grind another 10 microns, click it one more thing to the right, stop, take another picture, something like that. And if you've got all these pictures, you can then outline where your fossil is in all the pictures, and from all the stacks from top to bottom, you can set those and you can make a three-dimensional model, and yeah, bam, there is your fossil in 3 dimensions. The only problem is when you use this technique, your fossil is gone forever, but you have a beautiful picture of the insides of it. It's a trade-off. I don't know if you've read the book Blood Meridian. There's a guy in there called the Judge who's either like the first scientist or evil incarnate. It's kind of hard to tell, but he does a lot of this destroying and taking notes stuff. So you wouldn't always want to do this on a thing that you only had one specimen of. It's like less risky. Because if you mess it up and miss a couple of slices, your model's going to look like garbage and you can't go backwards. So make sure you get a good picture and it's not like blurry or out of focus every single time. But a lot of the best soft bodied fossils come from a place in England called Hertfordshire. And there's this one little deposit there of Silurian things, and they preserve all the soft tissue, and they're itty bitty. And they grow in these nodules, or rather, these nodules formed around them as they fossilized really quickly. And they are wonderful, but every single fossil that has been reconstructed from there is now gone forever. And so instead of a type specimen, which is normally the fossil that you preserve for posterity, they have supposedly preserved the digital scans of the fossil, which are a huge file, in some of these museums, like the Museum of Natural History in Oxford, I think, has most of these. And yeah, that's the idea. So that's yeah, high risk, high reward potentially. Most of those have been published in Nature, I believe, maybe a couple in Science, but that's pretty good going if you can get more than one Nature paper in a lifetime. Even if you can get one, that's pretty good going. Here's an example of a thing. So this is an arthropod, and look at this. As they're sectioning through this thing, they've got the eyes, they can read out the mouth parts, they can read out every little hair and filament on this crazy little swimming arthropod. It looks a lot like a group of things called Remipedia, just like surface, not paying attention to what the actual parts do. And I don't know, we'd have to look in the paper to see what they say it's maybe closest to. A lot of the problem you get with these studies is that because there is nothing else from before or after that has ever been reconstructed in 3 dimensions, and because a lot of these groups that they're reconstructing are interesting because they're totally gone and like unknowably gone, you'll get this situation where you could give it a name, but you can't even really say what it's related to because everything else is a smashed flat, hard parts only, partial fossil without all the interesting stuff that distinguishes these. So they have a real taxonomy problem, and maybe that's more a factor of what the authors feel about the world. They don't like to put names on things. I don't know, but it is potentially a pitfall of the process. But wow, look at that. And they're putting in the colors afterward as they are sectioning the model. It's a different use of the word sectioning. But yeah, here's one that we have done. This is one that some undergrads helped with in the lab, and it's a lot lower tech. What you can do to make this a little more robust is to mark on the surface of your fossil little increments. So if you pasted a little, bit of a ruler on there or something like that, then you could grind down like a quarter of a millimeter at a time, as long as you had something that was fine enough. And we just do this with sandpaper. So instead of setting them in resin or something, I normally just set them in plaster of Paris, because plaster of Paris is not as bad to breathe. Like breathing in all that resin is not good. Plaster of Paris is not great either. We wear a mask, but yeah. But anyway, you get all these little slices and you get this model, and the model disappears when it's sideways because the slices are thin and there's a lot of space between them, so it's just like you can't see anything. But if you tilt it just a little bit, then you can make out the little teeth, you can make out the little, look at that, dorsal median septum, the interbrachial area. And this was for a species where we had a description of the outside and we had a bunch of interiors that might or might not go with this. And so we found one that matched on the outside, sectioned the heck out of it till it was gone, and then we can actually tell which interior goes with which exterior. Yeah, so it's sometimes the only reasonable way to solve a problem, but it does result in destruction of the fossil completely and for all time. Yeah. Here's roughly the protocol if you want to try and do this yourself. You can do this in a program called GIMP. That's like a free version of Photoshop that's very good for cutting out the different parts that you want to reassemble. And then there's a thing called ImageJ. And ImageJ will let you stack up 2-dimensional images, and then we'll make a 3-dimensional projection out of these stacked 2-dimensional slices. And you can set how far apart the slices are until you get about the right proportions to make your model. So it's a lot of work, but it can be done, and the results are very interesting, I would say. Yeah, so what else can you do with this? Well, there's another technique, and the technique is our mascot for today. This is a CT scan. Like if you're going for a CT scan, sometimes they'll make you drink like a barium drink or a barium meal. It's just this thick pasty, gross stuff. But barium is going to show up differently in one of these CT scanners. And so you're going to be able to highlight the gut in this case, if that's what you want to do. There's other ways to label up the body, but that is a common one. But with fossils, like especially fossils that have been pyritized, like I think this one was, the fossil is already made of different minerals than the rock around it. We've seen this many times when they do those X-ray electron scattering sort of techniques. And so because of that, if your fossil is very different in mineralogy, like it's been totally replaced, but the soft tissue was preserved, and you probably won't know until you try it, then you can potentially just CT scan the thing with like a micro CT that sees really fine details. And here's a squid. Here's a look at, you can see its little eyeballs here. Here's one kind of sticking up, but it's hard to tell what it is. But yeah, scan that thing and they pop right out. And here are some of the tentacles at the front. Here, this is the tentacles. Here we've enlarged those tentacles. You can see the little break in the rock here. Here's one little sucker on the end of a tentacle, and it looks perfect. And that was just from a little micro CT. That's the little mu for micro computed tomography. So, interesting technique. And in some cases, it would get you to the same place as the serial grinding, but without having to destroy the specimen. However, if you have a situation where certain body parts are mineralized in one way and others are mineralized in another way, you may only see certain body parts. It's like you may get to see the bones but not the soft tissue parts, or the soft tissue but not the bones, or something like that. So there are potential drawbacks, potential reasons why you'd still want to serial section. And I guess the other drawback is that all these other techniques are kind of expensive compared to sandpaper, water, and a dream. But they all work. So there you go. Here's some study questions, and thanks a lot. We're going to try this in the lab. So get ready to serial section something.